Hi, how's it going? Thanks for joining. I think we'll wait for a few more people to jump on. Maybe we'll get started at a few minutes after 12. Hi, good afternoon, good morning, good evening. I know people are joining from all over. For those of you that have just hopped on, we're going to give it a few more minutes for people to join. The webinar is being recorded, so I'll make sure to post it on that LinkedIn. If you don't have the LinkedIn invite and you want a copy of the recording, please feel free to message me and I'm happy to share that with you. Okay, I'm going to go ahead and get started because I'm going to start with an introduction anyways. If more people hop on, that's wonderful. Like I said, this is going to be recorded and I'm happy to share it with anyone afterwards. So I'll go ahead and share my screen. Okay, and can someone just confirm with a thumbs up? Yes, you can see my screen and it's a slide. Okay, great, thank you. For those of you who don't know me, my name is Molly Clemens. I'm based here in San Diego. I work for Nexus, and Nexus is a distribution company based out of Boston. We're distributors for different imaging equipment based outside of the US. So in a way, we're boots on the ground here in the right time zone. What we do is consult with scientists here in North America. And we have these long-term relationships with customers where we're able to determine what kinds of imaging they need. And we see ourselves as consultants. So we help with not only finding the right equipment for you, but then also end-to-end setting up the equipment, setting up experiments, and doing the analysis. So what's happening next week, starting on July 10th, is a workshop that we're doing in support of San Francisco State University. They are working with Stanford University and University of California, San Francisco, UCSF. And the way it's set up, for those of you that are not a part of that workshop, is that there will be PIs and students that have a crash course in microscopy and have 2 weeks to solve a biological question. This webinar is a way to give them an introduction to the technology, as well as a little bit of a taste for those outside of the Bay Area if you're interested in this equipment. For those of you that aren't familiar with quantitative label-free imaging, to give you an idea of where it sits in the field of microscopy, it's in this sweet spot between phase contrast and fluorescence. With phase contrast, it's very easy. We're all familiar with bright field, but we have low resolution. With fluorescence, which is considered somewhat of a gold standard for microscopy, we introduce additional invasive sample preparation. So we get things like cytotoxicity, phototoxicity, and holotomography answers that problem with the solution of capturing these structures in their natural condition without labeling. So the way it works is by passing light through the sample. And so this gentle light passing through the sample pauses as it passes through, and depending on the refractive index or the dry mass of the object, we get a direct correlation with pixel intensity to give us dry mass. We're able to reconstruct these digital images to get these QPI images. If you're not already sold on QPI, based on not having to label your samples, not having phototoxicity, you should also know that it's extremely fast. You're getting these high resolution images, and then what you're able to glean from that is so much more than you could with a visual, bright field image. You're getting quantitative information because the digital images can be passed through specific software algorithms. So although the images are beautiful, the data that comes out of them is extremely powerful. The first system I want to present to you that we'll be using at the workshop next week, starting next week, is the Phase Holographic Imaging Holomonitor. So this little guy actually sits inside of an incubator. We can measure things like proliferation, motility, dose response, morphology, etc. It's compact, automated, label-free. Like I said, it fits inside a standard incubator. It can also be put in hypoxic chambers. We feel like the Holomonitor should be in every cell biology lab. It can quantify various cellular parameters. It can also be used to analyze cellular processes, so we can look at differentiation and response to stimuli. And afterwards, data can always be reanalyzed with any of those assays. So this is really great for adherent cells. And we imagine that, in the coming weeks, students will be able to play around with this, get really nice generated plots. They don't have to do any additional analyses, although the data can be exported to Excel. And so it just becomes this really powerful tool for studying those cell dynamics and morphology in real time. To give you an idea of the images, I have this video of an MCF10 breast cancer cell. As we watch this video, you can see the cells are dividing and the population is growing. The cells are moving in different speeds and directions. They're communicating with each other. They're changing shape and features. And like I said, it could be in an incubator or in a hypoxic chamber. So this long-term study of in vitro cell population is being done in a stable and physiologically relevant environment. To give you an idea of the dashboard that the system uses, you can see all the assays here. It walks you through setting up your assay once you click into one of these options. And because it was designed by Swedish researchers, you can tell the instructions are very clear, very easy to follow. And I think this is a very student-friendly system. I want to show you an additional video for dose response. And as we're watching this, you can see it's roughly 20x objective. you're getting a direct measurement of a dose response, and I'll show you what those curves look like afterwards. Us sitting here, we can see the difference between the drug-treated cells and the control, but more importantly, the quantitative information coming out of it can be used for papers, they can be used for, their thesis. This study was over 48 hours. And to give you an idea, they were treated with 10 micromolar etoposide, and the algorithm in the software immediately generates a dose response curve at the end. So the students don't have to learn R, they don't have to do any complicated data science. And this is what the dashboard looks like at the end of the experiments. This is all automatically generated. And you can play around with actually the count. You can look at confluence and volume and normalize it to starting. If you want to do any more in-depth analysis and look at a specific time point, maybe you see an anomaly on your graph, it's easy to analyze the images after the experiment. So just one more beautiful video from the Holomonitor. This video is actually a human DU-145 prostate cancer cell. It's undergoing apoptosis after exposure to the etoposide in vitro. And you can see that central cell, it just shrank and it broke up into fragments. Those are apoptotic bodies. Those are subsequently taken up by the surrounding cells. It recycles the cellular building blocks. And I think it's wonderful to highlight the fact that these are pseudo-colored images. So the color is digitally added afterwards, but this is a completely unlabeled experiment. So key points for those of you that want to use the Holomonitor: remember that this is useful for high-level kinetics information. And we consider this extremely complementary for Phasefocus and Tomocube systems. So we already have pretty packed schedules for using both the Phasefocus and the Tomocube. I imagine that many of you will want to take your samples and take a look at the cell quality and some of those higher level kinetics information on the Holomonitor. So I'm excited to have that available to you. In terms of what it's used the most for is proliferation, dose response, wound healing, single cell tracking. But the fun thing about these workshops is that we always get creative minds doing something innovative. So I'm excited to see how people dive deeper using the Holomonitor. Jumping and switching scales to the Tomocube, which is a separate system, you can see immediately from this image that it's a much different scale. So we're looking at 3D information in this case. And the HT-X1, which is the Tomocube system, provides 3D information using refractive index distribution. So we measure volume, dry mass, morphological parameters. And again, we can get stable long-term time lapses, but we can also get single point images, same with the Holomonitor. When we think about what this system specializes in and what it works best with, we think of live cells, primary cells, stem cell, neuronal cells. We can also look at nanomaterial delivery. And the important thing to note is that it's this really high resolution without any speckle noise. So we can also monitor multiple samples and thicker samples. So because it's 3D, we're actually getting a Z stack that's at a range of 150 microns. So that's important to keep in mind as well. This video that's playing kind of gives you an idea of the really, really high resolution. You can see the membrane flapping. You can see different details of subcellular structures. And how does it do this, right? So you actually have multi-angle light going through the sample. And this video of a cell death, I think, really highlights how we're able to, overcome optical resolution limits that you would normally get. So in this image here, I'm not sure if you can see my mouse, but for the optical NA, our synthetic NA is actually much higher compared to this physical NA value. because of the fact that it's a synthetic image. And the resolution of a holotomogram is going to be better than a fluorescence image captured at the same NA. If we jump into this video of differentiating adipocytes, what I want to highlight here is that images are they're not being diminished at all because of photo bleaching or any sort of drift. The system is extremely stable. So you can interpret your data at the end of your experiment in a quantitative manner without any conflicting information that would be caused by labeling. There's no limitations for your time-lapse setup. So in this case, we have a 72-hour experiment, but we have images from up to 48 days. The limitation there is really your media refreshing and your cells. I think these images are gorgeous. You can notice that there's no coherent noise in the image. There's no speckle, stripe, blurring, nothing like that. Just highlighting some of those subcellular structures that we're able to visualize. And if you're thinking about your experiments for next week and the coming weeks, these are things that you might want to quantify. So we're looking at nucleoli. Lipid droplets are extremely easy to look at. You can look at different shapes of the nucleus, the envelope, volume, area. You can kind of go on and on with this. And although it's label-free, we actually have 5 additional channels for fluorescence imaging if needed, if you wanted to do something, maybe a correlative study. I love this video. I've shown it, I think, before to some people in this group, but we have a 3D visualization of cell division. And what's important to note here is that there's a correction of the Z position by a laser sensor. So that's how you get that stable long-term time-lapse imaging. So what you could pull out of this is volume, dry mass, concentration, surface area, and sphericity, all based on the refractive index. I'm going to fly through this video and go into the Tomo analysis. This is just a screenshot to give you an idea of the software we can use with the Tomocube. So in the Tomo analysis software, this is actually just a basic measurement of the XY slice, but students will be able to dive into their images. They don't have to learn how to use ImageJ or other software. They can use the Tomo analysis software. I don't expect any of the students next week to be publishing Nature papers after 2 weeks, but I did want to give you an idea of what the pipeline looks like for the Tomocube. So from sample preparation all the way to publication, you get these gorgeous images. You're able to do your thresholding using the refractive index. You can get calculations. And then in this case, we had lipid droplet quantification over time. So the Tomo analysis software is helping us dive deeper into the images that are captured. And lipid droplets are one of the easiest things for us to quantify. I also just wanted to show you a publication, for example. Again, I don't expect this to come out of a 2-week study, but in terms of quantifying lipid droplets, this was a study where they were able to compare live hepatocytes versus lipid droplet formation in hepatocytes that had oleic acid treatment. This is a really straightforward example of observing fatty acid concentration and adipocyte differentiation in live cells. Let me just continue to slide 21. Okay, so again, this is for 22 days. We don't have quite that much time at the workshop, but I want you to understand the broad range of capabilities of this system. So over 22 days, you're not seeing any amount of drift. If you wanted to do throughout these 22 days correlative fluorescence, you also could. And you can also do 3D fluorescence. So key points for the Tomocube that I want you to remember. You can quantify single cell dynamics. You can also get those population level assays from other instruments and then zoom in on the Tomocube. And what it's used the most for is those analysis of the cell dynamics. So if you wanted to look at fluxes, for example, we had really nice images coming out of the MDL a couple weeks ago on yeast and different nanoparticles. You can also do really nice 3D observations of thick samples. The third system that I'm going to talk about before I hand it over to our guest, Jess Rickman from Phasefocus, is the Livecyte. And so the Livecyte is the third instrument that we're going to have at the workshop. And I like to think of this as sitting kind of between the Tomocube and the Holomonitor because it's kind of like a Google Earth system. You can look at the full population of cells, but you can zoom in to an extremely high resolution for individual cells. And so you're getting a ton of quantitative information. Again, it's an incubator system, label-free, so you can have those long-term time-lapse images without any photo bleaching, any cytotoxicity. There is an optional 7 fluorescence channels that can be used, and we can go up to 96 well plates for this instrument. The software is going to automatically generate a lot of the plots for you at the end of it, so it's very student-friendly. And why don't I show you here in the mitosis video? So this gorgeous video, we're looking at mitosis happening. You can see the cells balling up during mitosis. They get brighter in the phase image. So in this case, we can monitor proliferation of single cells. We can also look at the cell lineage. I'm going to play it one more time. We can also follow the morphological parameters of different cell families. There's 3 different objectives on the system. We have 4, 10, and 20x. You can just see, obviously, really, really fine detail of that cell behavior. Getting into some of the analysis you might want to do, this is an example of the quantification you can do of mitosis. So you're getting really granular information about 2 different cells. In this case, it's the chromosome alignment during mitosis. What we really like to stress about the Livecyte is that you're able to find heterogeneity in populations that you might have previously considered homogeneous. This is just one example of a way we can quantify mitosis. We're running a simple surface line plot through the cell, and you're getting these wonderful line profiles. The dashboards that you're going to get to play around with next week, this is just one example of mitosis. You get these wonderful plots, and what's great about them is that you can really narrow down to looking at exactly the question that you're asking. So you can decide which data is input into the plots. You can smooth the plots, normalize, change the type of plot, and all of these can be exported. What you can see at the top of this image is that we're just looking at the mitosis dashboard, but there's also proliferation, motility, morphology of the population versus the cell. There's just a ton we can do with this instrument. And it's really easy to play around with, and you don't have to spend a year learning R and R coding, no RStudio, no additional data software is needed, which is really, really helpful. When we zoom into that single cell level and we add that quantitative information, what you can see here is this really, really accurate quantification of cell movement. So it's tracking the cells in the same way based on the center of mass within the cell. So you don't have any more manual tracking that's usually pretty inaccurate, and it's very time consuming. And in this way, we're able to actually track the lineages over a long period of time without having someone sit there and watch. When we look at the dashboards for motility and migration, what I like to point out is the directionality. If you're doing something like wound healing, you're able to actually see the directionality of those leading edge cells. So all of these metrics are going to give you additional insights into your cell behavior. So we're going a lot farther than your typical confluency or proliferation I think what's really interesting about the motility and migration is that you get a confinement ratio. So essentially it's how straight a path the cell is taking. And it made me think a little bit about my own life, and if there was a confinement path for me during my PhD, how I might have behaved differently. One thing we're going to be using in the workshop next week is the T-cell killing assay. The automated analysis is actually going to be launched in September, so I'm excited to demo this with some of those early users at the workshop. At its current capability, we can watch T cells attack and kill target cells. The morphology of both cells is changing, as you can see. And the changes can actually be measured and analyzed using the Phasefocus software. The target cells here are labeled red, the T cells are unlabeled, and which is what you typically would want. And we can segment the T cells and target cells separately. The green is actually an apoptosis indicator. So we're watching cell death. And the strength of this assay is that once we have that cell death event, we can actually roll backwards. And what that means is from the cell death event, we can go backwards and see these interactions of how many T cells interacted with the cell. What are the number of death events, and what is that history that's going on before? And so you're getting an amount of T cell killing information that's not available on any other system. When I show it to you with the segmentation, you can notice that there's not a limitation by the minimum object size, so it's easy to differentiate that T cell from the target cell. And I think that this is going to allow for really, really powerful analytics in terms of these T cell killing assays that we haven't been able to see before. So excited to share this to you guys. I'm excited to see what kind of insights we come up with in the next couple of weeks and share them with the group. When I show you this dashboard, this is just an idea of some of the information we can get out. When I talked about that rolling back from the cell death point, we're not only looking at the number of T cell visits, but the amount of time that T cell was there, the interaction amount, and then the cumulative cell death. So there's a lot of really granular information here. So the key points, similar to the other two systems, I want you to keep in mind is that the Phasefocus Livecyte is for quantifying single cell dynamics amidst a population. So comparing those single cell heterogeneities against the population. It's used most often for growth and proliferation because of the fact that we can distinguish growth from proliferation. And Jess is going to tell us more about that. We can also look at motility, migration, and I encourage and I hope that we get to see some co-cultures in the next couple of weeks. Again, it's similar to that Google Earth. We're looking at the full population, zooming in and zooming out, and really getting these insights. With that, I want to hand this over to Jess Rickman. She's a technical application scientist at Phasefocus, and she's going to share a case study for us. So let me go ahead and stop sharing. And Jess, let me know if you're able to share.
Yes. Oh, if you can, so you've disabled participant screen sharing. If you can.
I think I made you a host.
Okay.
So let me try again. Let me do it again. I'll make you the primary host, change host. Let me see, did that work now?
Yes, we're all good. Thank you.
Great.
Excellent. So, can you guys see that? Is that all okay?
Yes, I can see it. Not in presentation view yet.
There we go.
Perfect.
Awesome. Thanks, Molly.
Thank you so much.
And yeah, thank you so much for giving us a great introduction there. I completely relate in terms of the confinement ratio with the PhD. I'd say my meandering index was all over the place. But yeah, so hi everyone, thanks for joining us today. I am from Phasefocus, the company, and we sell Livecyte, our microscope. As Molly said, earlier, we do various wide-ranging applications. So mostly focusing, well, there's loads of things that we focus on, but a lot of the researchers that we work with are doing things such as the growth proliferation, as well as motility and migration. Those are kind of the bread and butter. But it's great to use those, and it's great to have that option of just using label-free imaging. You don't have to add any fluorescence, and you don't have to kind of, that doesn't lead to any kind of potential phototoxicity. But fluorescence is also a really useful tool when it's managed and handled well. And so I'm going to go through at the moment how we can use this fluorescence in terms of identifying cells in a co-culture. We'll look at very briefly the software. I'll lead you through just some of our dashboards and show you how easy it is to use and how we can get information out of co-culture experiments. And finally, I'll talk to you about one of our case studies, which is the Huntsman Institute, which is in Utah. They're studying melanoma and looking at the causes of progression of melanoma from kind of benign states to more malignant states. So to start off, as I was saying, fluorescence is really useful in terms of imaging because it can allow us to very easily identify different cell subtypes within a co-culture. So you can have cells which are labeled in one certain way, and we can draw those out and compare those with cells which aren't labeled. I'll just hide this. Sorry, there we go. So this is what we did in this experiment here. We looked at fluorescent cells co-cultured together with label-free cells and tried to draw out metrics in terms of their proliferation and growth, motility and morphological kind of behavior. And with this time-lapse imaging, we could see this changing over time as well. So what we wanted to do was to compare the isolated cultures, so the cell subtypes growing on their own, and compare that behavior with how the cells acted when they were together and able to kind of interact and signal with each other. So this video is just giving you a little lowdown of the layout of the experiment. On the right-hand side, we've got the labeled differentiated cells. These were labeled with mCherry. And then on the left-hand side, the progenitor cells, which were label-free. On the far left of the experiment, we then had the co-culture. You can very clearly see which cells are which, and so is the software. So we can form gating, which enables the software to identify which cells are which. And from there, you can apply it to our dashboards and look in terms of the data analytics at what is happening. So I'll head off here and go on to our dashboards now. So Molly gave you a bit of a lowdown about what all of our dashboards are, but essentially Livecyte has got lots of potential analysis tools to be using. You can go very in depth if you want to, but you can also, with the dashboards, have a really good overview of all of the different metrics that are collected during your experiment. And I should say one experiment will give you all of these metrics if you want it. So you can access it, even if you're not specifically looking at it, you can see how behaviors change and correlate and potentially see things that you wouldn't otherwise identify. So this is the experiment that we have. And I'm just going to play it. And we can see, hopefully, if my internet's good enough for you guys, that we have the red fluorescence flashing on and off during the experiment. So this is because we did intermittent fluorescence imaging. We imaged 3 phase images for every 1 fluorescence image. And this is just a way that we can mitigate and reduce that phototoxicity. So we don't expose the cells to as much radiation. What the software can do is, even when it is on a label, a non-fluorescence image, for example here, it can still identify which cells fluoresced in the previous frames and tie that back with the analysis. So it will still identify those cells as fluorescent, and within our fluorescent gate, even though there's no fluorescent image for this frame. So let's take a look at some of our dashboards. We're looking just now at the two isolated cultures. So the brown are differentiated M-cherry labeled cells, and the green are the label-free progenitor cells. And we can see there's some differences when they're grown in isolation. We have our cell count and our cell doubling time. From this, we can see that our progenitor cells are growing slightly faster than our differentiated cells. Similarly, if we go on to the random motility, we can see that our progenitor cells are also slightly faster than our labeled cells. It's just a nice show of how easy it is to smooth your data, make it more easy and more presentable. So this is great. We can see the two isolated populations, but what if we want to have a look at what the cells are doing in a co-culture? If we add this in, we can see that it's kind of slap bang between the middle, because we've got a mixture of the two cells. So we can't really discern what's going on in terms of the individual cell subtypes. So what we need to do is we need to apply a gate. And because we're kind of a bit short for time, I've already made this gate up for you. Essentially, we sort our cells similar to a flow cytometry software. We sort them here based on the red fluorescence, and then we make a gate which captures that red population of cells. And we can see here quite nicely, we've got those outlined cells are the ones that are captured by this gate. So if we head back to our dashboard, we can then apply this filter. We'll just look at the isolated cells, at the co-culture cells, sorry. We'll apply this filter and look at the cells that are both inside the gates, so the red cells, and outside the gate, the label-free cells. Okay, so there's a few changes that we can see. If we go back to the proliferation, we can see that that difference in terms of the cell doubling time is actually much smaller than it was before. If we normalize to the start of the experiment, we can also see that these curves are much more similar. And so that change and that difference in proliferation levels is kind of is shrunk quite a bit. Similarly, with the random motility, we can see there's a little bit less of a difference in terms of the speed of the cells, especially you can see it quite nicely here. Previously, we had two very distinct lines, whereas here these differentiated cells towards the end of the experiment are actually moving at a similar velocity to the progenitor cells. So it's just a great example of how cells behave in different environments, and the importance of being able to kind of preserve and conserve these cell signaling pathways that can lead to cells acting in a different way. And obviously in biology, we want to make sure that we have experiments that are as physiologically relevant as possible. And so to be able to grow in these co-cultures and to be able to still identify and analyze these cells when they're grown together is a really valuable tool. Okay, so now we'll have a look at the Huntsman Institute's data. So they were looking at melanocyte plasticity. This work is based off of a paper that was published a few years ago, a couple of years ago now, and it was looking at how melanocytes progress through from kind of healthy forms through into melanoma. There's also some additional work that they've been doing since, but I'll talk briefly on that at the end. So as I was saying, melanocytes are really adaptable. They can be in lots of different states. If we look at the schematic here, we have the healthy melanocyte just on the left-hand side, and this can transition from a healthy state through into a hyperproliferative state where it can then rapidly divide and form moles. So everyone has them on their skin, little brown moles. Sometimes they hang around for your entire life and they do nothing. but sometimes they start rapidly proliferating again and can cause significant problems and melanoma. So BRAF V600 is a mutation which has been seen and is highly relevant in this incidence. So BRAF V600, I think, is been seen to be present in 80% of nevus or mole cells, and is present in 50% of melanoma cases. So it's highly relevant, and it's very likely that this mutation is doing something in this process. So they wanted to look at this secondary stage of how cells can go from a benign nevus through into a melanoma, and how this can be controlled and potentially use this to treat and prevent melanoma. So they used Livecyte. They wanted to track and characterize their cells, and basically to be able to really dig into the mechanics of their cell biology and understand more about the interactions of these different proteins. So they started off with a experiment which gave them a result that they expected and one that they really didn't expect. So they created an inducible BRAF V600 mutation, which was induced by treating the cells with doxycycline. So on the left-hand side, we can see that the cell count, as the dose of doxycycline is increased, the cell count and the level of proliferation was reduced. So this is the higher doses, and we can see that the cell count is going up much slower than these controls. And this was expected. They'd seen this before in previous literature. But what they weren't expecting to see was this graph on the left. So because with the QPI imaging, you're able to measure the dry mass of a cell, you're able to see not only how a cell is dividing, but you're also able to see how much your cell actually weighs. And what they were seeing was that even though these cells had stopped growing as quickly, they were still, stopped proliferating as quickly, they were still actually growing. And so this actually went against previous theories that had been proposed as to what is actually happening when a cell is in a stable state in a mole. So previously they thought it was oncogene-induced senescence, whereby there was cell cycle arrest at the G0, G1 stage. But actually, this data indicates that this is not the case, and that it's actually at the G2M stage, because the cells are still growing. This oncogene-induced senescence theory suggests that cells don't actually grow and they just do nothing, but we can see that this isn't the case. So this is data that you just wouldn't be able to get with any other imaging other than QPI. And previously, although there had been kind of papers where they theorized against this oncogene-induced senescence, no one had actually suggested or proposed an alternative mechanism. So this was really novel research. So next up, they decided to zoom in and look in more detail at a downstream mRNA, downstream from the BRAF gene, BRAF mutation. And this mRNA was called ORC-B. And they basically managed to identify microRNA which controlled this mRNA, this ORC-B. So essentially, the process goes that the BRAF-V mediates the activation of this ORC-B mRNA, which then leads to this hyperproliferative state. So this is how we get from our stable moles through to our melanoma. This microRNA, however, controls the ORC-B mRNA, and essentially sends the cells back into their stable state. And this is fine. You can have a mole, it will be there for the rest of your life, and it will never do you any problems. However, the issue arises is when you get mutations in this ORC-B, and this then frees the gene from the microRNA control and allows this progression towards a melanoma. So what the team at the Huntsman did was they transduced the ORCB mutation into melanocyte cells. So they got primary mole cells and transduced them along with mCherry, so it was linked. So the ORCB expression was kind of signaled by this mCherry fluorescence. What they were able to do was they were able to track the mCherry expressing cells and compare the proliferation levels to the non-mCherry expressing cells, so those cells without that mutant. And they saw that the mCherry cells divided significantly more than those non-mCherry expressing cells. An important thing to note here is that they were using primary cells from moles, and they had a very small sample size. Rob said they had something along the lines of just a handful of 96 plate wells filled. And so this is potentially extremely difficult to get reliable data from. But because Livecyte is basically looking at each individual cell and treating each individual cell as a data point, they were able to get statistically significant data, which they then published. So they're doing more work now. They're basically looking at how differential levels of fluorescence and the change in levels of fluorescence over a cell's lifespan impacts the behavior of the cell. They're also looking at additional screening of different compounds that can be preventing this process of melanoma from occurring. And I think they've got some defense funding for this research work. So just to summarize, they were able to look at this growth of the cells independently from the proliferation, and from that they were able to draw out novel new potential mechanisms to understand these processes. They were able to image their cells over long periods of time, which was a non-phototoxic imaging of primary cells, and that allowed them to basically monitor these cells and see how they changed. And they were able to produce reliable data from very small levels of sample because each cell was treated as a data point. If there's any questions or anything like that, then you guys go for it. But if not, then Molly, I can hand back to you.
Yeah, thank you so much, Jess. And I'll give it a minute in case anyone has any questions for Jess. I'm curious what the longest time lapse that the Huntsman Institute was doing with some of those cells.
Ooh, so I think it was something along 5 days. So they had yeah, it was I think it was 5 days they got going. But they were doing kind of experiments where they were taking their cells in, putting them in, imaging them for 5 days, and then taking them out, and then on like 10 days later they were then putting them back in. So it was really long periods of time that they were imaging over, but because of yeah, they kind of did it in chunks of 5 days. I think you can use it for longer, but that's just
yeah, I was just thinking about like the average age and behavior of a melanocyte and those types of things. But yeah, thank you so much for presenting that. I think that it's really important to remember that the difference between growth and proliferation really shines in studies like that, and how we can kind of go down the wrong path if we're not able to differentiate those two.
Yeah, definitely.
If there aren't any other questions for Jess, one really practical component for the workshop next week and the next coming weeks is to remember the different plates that are compatible between the 3 systems. And if you wanted to do an experiment that incorporated all 3, our recommendations would be to use either the Sarsted 6-well plate or to choose one of the Abiti plates that goes across all 3 of the platforms. Otherwise, you might have to replate your samples, and we don't want you to have to do that. If you have any questions about your sample prep, about compatibility, anything like this, please reach out to me. I want to make sure that everybody is prepped and ready to go, and you know, that we have all the correct plates. In addition to that, if you are using the Livecyte, just remember you can use different coatings because they are adherent cells, but for Livecyte, you can use Matrigel, agarose, cell-derived matrix, fibronectin, and if you plan on using 20x or higher, Please make sure you're using a thin plastic bottom if you do have to use plastic. However, glass is better. You guys know that. The brands to kind of stay away from that we've had issues with are the Maytech and the Griner plastic bottom plates. But again, that's a lot of information to put out there. So if you have any questions about it, please touch base with me. Otherwise, Jess and I will both be there on Monday if you have questions, and so you can come talk to us in person. With that, and I want to leave some space for questions. The takeaways, the overall high level, what I want you to think about for your experiments is that this Holomonitor is your workhorse. It's your, look at your cell quality. You can look at cell population kinetics. If you want to explore that population scale data and then also zoom in on a single cell, let's try out the Livecyte. And if you need to look at anything subcellular or you want to look at 3D cell-to-cell interaction, let's try out the Tomocube. But I'm excited to see what everybody brings to the table and to see what the students come up with. And with that, I'm going to go ahead and I'm going to stop sharing my screen. And if there's any questions from the people on the call right now, otherwise, the webinar will be recorded. I'll leave it open for a couple more minutes in case there's any other questions, but feel free to hop off. And thank you so much for joining us and for listening to these. I hope I get to see everybody in the next couple weeks for the workshop. And thank you again, Jess. I really appreciate your presentation. That was really, really insightful. I'm looking forward to seeing you next week.
Thanks, Molly. Yeah, really looking forward to it.
Cool. All right, well, take care, everybody. Thanks for joining.
Bye.
Go ahead and end it. Yeah, take care. Thanks.